Hello everyone, welcome back for more biotechnology. Last time we talked about polymerase chain reaction, we talked about the theory behind it and some of the practical aspects of it. So let's talk about taking it a step further and how we can further use applications of PCR technology to get certain things done in the lab. The first thing that we're going to talk about is a super duper application of PCR called QRT-PCR, or Quantitative Real-Time PCR. So QRT-PCR is a type of PCR application that is not only going to give you the DNA that you want, but it's actually going to quantify how much template DNA or RNA is present in a sample based on the speed at which amplification occurs. So in addition to all the reaction components that we talked about before, all the different goodies that you need to mix up in the tube, we're also going to add in a fluorescent dye. Usually we're talking about something like cyber green. So that fluorescent dye is going to intercalate in duplex DNA or RNA when the product is formed. So the idea here is that as the thermal cycler cycles back and forth and continues to generate more and more template DNA, you're going to have more of this fluorescent dye that sticks into the DNA and is then quantified and detected based on fluorescent absorption. So this type of application usually is going to require a special type of thermal cycler that not only can change temperature rapidly, but can also shine fluorescent light on the sample and detect how much fluorescence is absorbed. So the basic principle that we're applying here is that if the amount of original template DNA is very, very, very low, it's going to take more and more and more PCR cycles to amplify it up to a certain amount. Whereas on the other hand, if the amount of template DNA is much higher, it's not going to take quite as many cycles to get up to that same amount. So the real data that we're interested in here is something called cycle number. The cycle number gives you an indication of how many cycles of the PCR reaction it took in order to get us to a certain point on the fluorescence scale on the y-axis. So looking at this is going to give us some kind of approximation in terms of how much template was there originally. Usually the most common application of this is detecting the amount of messenger RNA in cells. So you can get a good idea of how much or how little a gene is being expressed in a cell by using QRT-PCR to determine how much of that messenger RNA was there originally. Okay, so now let's talk about something a little bit different, DNA fingerprinting and, in general, forensics. So forensics, we're obviously talking about the application of different hard sciences like biology and chemistry and things of that nature to solve crimes. Usually kind of the most famous or most common things that you see on TV shows is you're talking about solving murders, right? So the basic idea behind using science and forensics is that, especially when you're dealing with DNA, the idea here is that because all cells contain DNA and many, many crimes are going to involve criminals leaving behind blood or other bodily fluids that contain cells and therefore contain DNA, we can use PCR technology to amplify what little DNA is there, and we can use other things that we're going to discuss here shortly to identify a perpetrator. So the idea here is that, okay, so as humans, even though we have little differences here and there, humans share more than 99% of our DNA sequences. We're really not that different from each other when it comes right down to it. But oftentimes there are going to be enough differences from person to person to allow us to identify a suspect based on any DNA that is left behind at the crime scene. Now, this isn't even to mention other identifying factors like your blood type, fingerprints you left behind. So there's a lot more that goes into forensic science other than just DNA. So if you're interested, I definitely recommend checking it out. But we're going to focus our discussion here specifically on DNA fingerprinting. So DNA fingerprinting refers to the method of identifying a suspect based upon DNA evidence that is left behind at the crime scene. So we are going to use conventional PCR in the way that we described previously. But what we're going to actually be looking for here is something that all humans contain in between their genes in regions called microsatellite DNA, and these stretches of DNA are called tandem repeats. So tandem repeats are not gene sequences. They don't code for protein. They're mostly meaningless, but how many tandem repeats you have in certain stretches of DNA kind of tends to vary from person to person. So one person may have a few of them, and the next person may have a whole bunch of them. So there are PCR primers that are available that will anneal to different places on different chromosomes and should amplify regions that contain different or varying numbers of these tandem repeats. So, for example, if you have DNA evidence left behind at the crime scene, which is here on the right, you may, when doing the PCR analysis, you may get 1, 2, 3, 4, 5, 6 different PCR products that kind of run the gamut in terms of how big they are. So the one on top and the one on bottom will be very small products. The fifth one here will be fairly large, and then the 3 in the middle here should be somewhere in the middle. So if you identify 3 different suspects and you take DNA samples from them and you subject those samples to PCR analysis, you should be able to, at minimum, eliminate suspects that do not match or do not fit the DNA left at the crime scene. So if you'll notice here, suspect number 1 is not really a good match because the size of the fragments that are left behind, the size of the fragments that are amplified by the PCR primers, does not really seem to match what is in the crime scene DNA. Suspect number 2 looks like a match for the first tandem repeat, but doesn't really hold up for some of the others, especially the second one that you see here. This one seems to be a little bit too long. And then suspect number 3, again, doesn't really seem to be a perfect match because some of these do not seem to be matching up very well. So if you run everything out on a agarose gel, you'll notice that you get, like I said, 6 different fragments from the PCR reaction based on where the primers anneal to different parts of the chromosome. So the DNA left at the crime scene should produce PCR products that run from very large to very small. And then based on the banding pattern of the DNA left behind by the suspects, you should be able to kind of tell who matches up the best. And it looks like I was looking at suspect number 2 in the wrong way. This one right here seems a little bit longer than this one right here, but everything else does seem to match up pretty well. Maybe my eyes are just playing tricks on me. So based on the agarose gel, suspect number 2 does definitely seem to be a pretty good match with the DNA left at the crime scene. So that is actually going to do it for this very short video. There are a number of other PCR applications that we could talk about, but we're not going to get into that today. So definitely, if you're interested in PCR, I definitely recommend you stick around for more biotechnology classes in the future because there is definitely a lot of interesting stuff to talk about there. So we'll go ahead and wrap up this video for now. Our last video in this chapter is going to focus on DNA sequencing, yet another application of in vitro DNA synthesis. So thanks again for your attention, and I will see you next time. So long.